Okay, hello. I'm back with an update on my research question: how can we use CA models to simulate the growth of neurons and their interactions in the human brain? So, unfortunately, for this presentation, excuse me, sorry, notification. Um, uh, Unfortunately, I wasn't able to have a finished product again. So I'm gonna kind of walk through my code and the updates I've had since the draft, in hopes that by Friday I'll have a polished and finished product and product no, project. And obviously I can make another presentation if needed, but I thought that this would be okay considering that I put a ton of hours into the code that I'm working on. But anyways, so I'll just start off with basically since last time I was working on implementing here. I'll just show you. So basically, since last time, I was working on implementing. No, I finished implementing, and I had implemented this a basic dendritic growth um, pattern tree, where here in um, oops, here you can see that you have a bunch of just neurons and axons, and then the blue is all the dendrites. So basically, what happens is a random when a neuron is born, this in this simulation we're assuming that they are all instantly born at the same time in different random locations. Obviously not what happens in real life. I wish, but it basically does have a random walking implementation that connects between two valid points. Valid points being the beginning of a dendrite and up the dendrite end, which is basically where there's no other type of cell. Um, so yes, and since then, let's see. Since then, I've had a few ideas. I had a few ideas that I had outlined in the draft that I had plans on implementing, and um, I have implemented basically environmental factors. So that's amniotic fluid pressure, cortical pressure. That obviously influences how neurons migrate and grow. Different types of neurons. So if you look, not specifically not neurons, but cells. If you look here, um, so when a baby's born, they start off with a bunch of stem cells that are said to be like totipotent or pluripotent or multipotent, which pretty much means varying levels of what they can differentiate into. So if something is totipotent, that means it can differentiate into any type of cell in the human body, any type of stem cell. Um, so a neural stem cell is a type of stem cell that starts off in the brain and has ingredients to the blueprints, or the blueprints for the blueprints to create neurons, neural progenitors, glial progenitors, all of this stuff, specifically neural progenitor and glial progenitor. But what a neural stem cell can do is it can say, okay, depending on the environmental factors present at the time, it can either uh, reproduce itself, which early in brain development, the majority of time, neural stem cells are self-renewing, especially towards the center of the brain, and obviously neurons are created very rapidly too. But, anyways, um, so these neural stem cells can either self-renew or they can differentiate into a glial progenitor or differentiate into a neural progenitor. Now, normally what happens is you'll see a neural stem cell producing one of itself and one of a neuron or a neuronal progenitor. So I implemented this tree right here. I don't. Hopefully you guys can see my cursor, but the tree from neural stem cell to neuron. I have not implemented oligodendrocytes or astrocytes yet, which I plan on doing for Friday. Um, but yes, and then where am I? So I've also implemented cell division in neurons. Yes. Precursor cells. Yes. Neural growth rateally increases followed by a tangential increase. So I have implemented this, which is pretty much the idea of how I'm trying to model early developmental brain growth. So brains, um, they go radially outwards, right? The cells divide and go outwards based on the nutritional availability in the brain, how much resources a cell has, the pressure in the brain at that particular position, and the density of other neurons around it. So neurons will, or the brain will grow outwards, and then it will grow tangentially and fold inwards, which causes us to have folds in our brain and to have, you know, cortexes, our cerebral cortex, are different regions of the brain. The prefrontal motor cortex. All those different regions happen because of the tangential growth. They start from the tangential growth along the surface of the brain. So, so I also implemented gene expression in intrinsic weights because obviously, all cells, aren't encoded with the same exact DNA. I mean, there are some tendencies for some cells to do one thing or some to another, so I implemented that. So I'll go through the code now. This hasn't changed. This is just to find a connection point, and now it's just simply the magnitude, or straight line path. And in that straight line path, the straight line path is modeled, and then it chooses. What it will do is I'll just show you. What it does is, actually, I use it somewhere for the axon, but it's not very complex. It just finds a straight line path and connects the dots. But here, in my place dendrites, or update dendrites, this is where I implement my random walker, where there is a target synaptic end and there's a synaptic beginning, or the starting index ij, and it'll take a random walk while going towards the target position, or the dendrite end, while avoiding valid neighbors. Now, this isn't the most realistic implementation because in real life the dendrites would be growing at the same time as everything else, but I'm going to say that the dendrites grow at a much faster rate because partially that makes sense, partially it makes no sense at all. But hopefully I'll make it more realistic later. And then I implemented a new class called the neuronal stem cell. So this is mainly where the agent-based modeling that I've implemented now have implemented in this project comes in. So I have each neuronal stem cell has a certain amount of energy, differentiation potential, and intrinsic probability. So every time step it consumes energy, obviously, and there are chances based on the environment. Each cell is given at each time step a differentiation chance. So depending on its energy levels, depending on its genetic, if genetically that cell wants to differentiate fast, it's able to go through its cell cycle fast, or we have an environment modifier that will change the which is where sorry. Anyways, what I was saying was, the neural environment, which is another class I made, starts off with random growth factors, nutrient density, inflammation levels, which my way of saying cortical pressure, amniotic pressure, buildup, density, combine into one factor. Now, this is also not as realistic, but again, I'm trading off between complexity of my simulation and accuracy. So the local factor is just is an equation just based off what's going on around that cell. So depending on the environmental factor, or the environmental modifier and genetic modifier, it'll have a complete differentiation probability. And then there's a differentiation type. Now, this one was slightly more tricky, but basically what I'm modeling here is the growth of the brain radially outwards. So what I have is that this distance factor. Now, the distance factor, this distance factor, basically, I'll rerun the simulation again, but say you have the center, right here. Hopefully you guys can see my cursor, but if you have the center, as the cell increases radially from the distance between it and the center, whatever cell that is, the distance factor increases. So if I go back up here, that means that, okay, 1 minus distance factor, that means that as the cell is further away from the center, it becomes less likely to self-renew and more likely to become a neuron, which is just a slightly skewed way of, biologically skewed way of getting it to display the relationship that is realistic. But it's a good way of doing it because it models the correct result. And then if it does self-renew, it takes a bunch of neighbors, determines if it's a valid neighbor, and then it will split into two. So it splits into two and says it'll become a neuronal stem cell and then another neuronal stem cell that's placed at another distance. Now, one thing to note is that I am assuming that when the birth of a new cell happens, they start at the same exact energy as the very first cell. So again, it should be it's starting with full energy basically, instead of, it should technically be different from that, but I haven't modeled that portion yet because that's another complex model variable. So the neural progenitor does the same thing. And yeah, I mean, I don't want to bore you guys too much with code. But consume energy is this. The GLA progenitor again is a placeholder, and then there's a mutation rate. So there's a 1% chance here that a genetic marker changes. So that would be like something, a chromosomal, like a DNA shift. So that there's a variety of factors that could play into that. And in the sake of time, I will just go over that in the report. I don't know how long I've been presenting. Oh, way too long. Oh my god. I'm so sorry. But then it just displays it. So let me just run it. So as you can see, at first it starts just producing neural stem cells, and then there's the dendritic growth. And then as you can see, it seems like in this simulation, which is similar to last one, the nutrient density seems to be up in this top left-hand quadrant. So you'll still get some over here, but you see a lot of dendritic growth and neural growth up here. I think that's just because of the way that the environment is selected or randomly generated. But it'll just go through this, grow the dendrites and the neurons, and etc. So this is definitely not the prettiest way to visualize it, considering the amount of complexity in the model, but that's a work in progress. All right, thanks.